Today topic is fungi general characters, and we have seen many questions coming from this section, and especially I have seen two questions of 4 marks in December 2014, and same question was repeated in December 2013. Fungi has in present classification, fungi has been divided into 8 kingdoms: Chytidiomycota, Zygomycota, Neocalliomystigomycota, Microsporidia, Glomeromycota, Blastocladiomycota, Ascomycota, and Basidiomycota. In earlier times, the classification was Phyco, Asco, Basidio, and Deuteromycetes. So, Phycomycetes has been removed from recent classification of fungi, and instead there are classes like Chytidiomycetes. This was the family of this one, and different orders has been upgraded up to the kingdom level. So, Chytidiomycota, it is predominantly aquatic, saprophytic, parasitic, and have posterior flagellum. Zygomycota, most are saprobes and reproduce sexually and asexually. Neocalliomystigomycota, predominantly located in digestive tract of herbivorous animals, anaerobic, terrestrial or aquatic. Microsporidia, all obligate parasites like fish, crustacean, human. Microsporidia can grow and reproduce only inside host cells, and they lack functional mitochondria, so dependent upon their host for their energetic needs. So this is also applicable for Glomeromycota. They cannot survive outside the host, but these are not parasites. Glomeromycota, these are arbuscular mycorrhizae present in terrestrial with a small presence on wetland. Reproduction is asexual but require plant roots. Blastocladiomycota. Blastocladium means offshoot fungus. Blastocladiomycota along with the Chytidiomycota and Neocalliomystigophora are aquatic fungi that produce flagellate zoospores. Ascomycota, sac fungi, and Basidiomycota, these are the bracket fungi, toadstools, smut and rust, and they have sexual reproductions. This is the configuration of cell wall in different division of fungi. The oomycotes have mainly glucans and cellulose. Chytidiomycotes have chitin 58% and cellulose is absent. Zygomycota have glucans 44% and chitin 9% and cellulose is absent. Ascomycotes have 39% chitin, 29% glucans and cellulose is absent. Basidiomycotes have 50% glucans and 30% chitin. Cellulose is absent. So cellulose is present only oomycota. Now kingdom protozoa. Protozoa includes Acrasiomycetes, Dictyosteliomycetes, Protosteliomycetes, and Myxomycetes, and Plasmodiophoromycota. Plasmodiophoromycota contains Plasmodiophora, which cause club root disease of crucifers. Kingdom Staminipela or Chromista. It contains Hypochytriomycota and Oomycota. Saplognathales, Pythiales, and Pernosporales are the orders of Oomycota. In December 2013, there was question: fungal group presently classified under Protestes, Zygomycetes, Oomycetes, Deuteromycetes, and Discomycetes. The answer is Oomycetes. Oomycetes comes under this one. Oomycota is the different thing, and Oomycetes is the part of protozoa. Kingdom fungi, Eumycota, Chytidiomycota, Zygomycota, Ascomycota, and Basidiomycota. These are the division of kingdom Eumycota. Now, Ascomycota comprises Arcascomycetes, Hemiascomycetes, Lactomycetes, Hymenoscomycetes, and Pyranomycetes. Pyranomycetes has some orders like Erysipelas, which cause powdery mildew, Pagagales, Pagaia fungi, Halotiales, means Tephorina, which cause rust of turmeric, and Lichenorales or lichens. Most of the fungi forms lichen, and then Loculoascomycetes. Basidiomycetes comprises two families. Holobasidiomycetes means agaricus, and Heterobasidiomycetes means rust and isthmus. Heterobasidiomycetes have two orders, two families, Uridinales, Puccinia graminestrici, and Astilaginales comprises Astilevo, that is isthmus causing fungi. Slime molds. Slime molds differ from Eumycota in phylogenetic, physiological, and ecological point of view. Their vegetative state may be cellular slime molds or plasmodial slime molds. Cellular slime molds resembles amoebae, and plasmodial slime molds, this may be cynocytic plasmodium, multinuclear single-celled plasmodium. Plasmodial slime molds have motile stages with two anterior whiplash type of flagella. Okay. Chytidiomycetes have single posterior flagella. This may be the difference. Nutrition in slime molds is much different from Eumycota. Eukaryotes show extracellular degradation and absorption, whereas in this slime molds, they have animal-like nutrition. Now, kingdom Staminipela. They have combination of photosynthetic and non-photosynthetic plants. Formerly, they were called as chromists. Some have photosynthetic apparatus, such as the covering of chloroplast in endoplasmic reticulum, also found in Fungi, and absence of chlorophyll B means chromatophore. Kingdom Chromista Staminipela currently includes the diatom, golden and brown algae, chrysophytes, and cryptomonads. This table shows difference between Staminipela and Eumycota. As in plants, inner mitochondrial membrane is folded into tubular cristae. Eumycota, mitochondrial cristae are generally lamellate. In Staminipela, dictyosomes are present. Eumycotes have single cisternae. Golgi apparatus is much reduced like single cisternae. Now, important point is the lysine synthesis. Staminipela has two pathway of single pathway of lysine synthesis via alpha epsilon diaminopimelic acid pathway, whereas in Eumycotes have AAA pathway, that is alpha amino adipic acid pathway. Protozoans are auxotroph. Lysine is synthesized by alpha aminoic acid pathway route. This has been repeated. Chytidiomycota, most chytids grow aerobically in soil, mud or water and reproduce by zoospores. Zoospores is the most important characteristic of chytids. Chytids have a single whiplash type of posterior flagellum. Thallus may be holocarpic and eucarpic. In holocarpic thallus, complete body is converted into reproductive structures, whereas in eucarpic, a part of the thallus is converted into reproductive structures. Eucarpic thalli may have one, which is called as monocentric, if many, polycentric is pharyngia. Zygomycota, they lack motile stage. Asexual reproduction by aplanospore or sporangiospores contained in sporangia, and these sporangia may produce on sporangiophores in rhizopus and mucor. Sexual reproduction is by isogamous gametangial copulation. Gametangial copulation means entire gametangia fuses. Formation of zygospores. Cenocytic mycelium cell wall has chitin and chitosan. Chitosan is the deacylated derivative of chitin. Ascomycota. Sorry. Erysipelas, the causative agent of powdery mildew disease. Lobulobaniales. These are the harmless ectoparasite insects or parasites on insects. Mycelium is cenocytic, perforated septa, free movement of organelle like mitochondria and nuclei across septa. The one important cheese is proteinous organelle, Voronian bodies found near the central pore, near the perforation of septum. Basidiomycota. Basidium is a spore-bearing cell producing basidiospores externally. Basidium is the spore-bearing cell and it produces basidiospores externally. So, difference between basidiospores and ascospores. Ascus are produced within the sporangium, and basidiospores, these are just like conidia. They are produced outside of this basidium. Mycelium is septate in rust and smut, but in agaricus, dolichopore septum is found. Mycelium is not cenocytic. It may be monokaryotic and dikaryotic in Basidiomycota. Next is the Glomerulomycota. Members of Glomerulomycota forms orbuscular mycorrhizae with the thalli of bryophytes, roots of vascular plants. Glomerulomycota have generally cenocytic, sometimes septate mycelia. They reproduce through blastic development of hyphal tips to produce spores. This is the important character. They produce spores from their tips, and these spores are called as glomerospores. Glomerulomycota has one exception, Geosiphon pyriforme. It is the only example of non-mycorrhizal species within the genus Glomeromycophyta. This slide has slight mistake. This Geosiphon should be here. One thing. It is the Geosiphon pyriforme. This is a species of geophyte. And this Geosiphon form inflated bladder cells that contain nitrogen-fixing cyanobacteria, that is nostoc punctiforme. Glomeromycota, like microsporiae, they cannot reproduce out of the living host. Now, this is the question in December 14. Following the table, and in the left column, there is the fungal groups, and in second, that is the characteristic. No regularly occurring septa in the thallus means that should be the zygomycetes. Perforated septa, either basidio or ascomycetes, depends what is the type of perforation. And forms arbuscular mycorrhizae and on root plants. So most likely it will be glomeromycetes. Have juice spores with flagella. Juice spores is the peculiarity of chytridiomycetes. So what should be the answer? Have juice spores with the flagella means last one D should be the chytridiomycetes. So we have in this one. And now this one A means chytridiomycetes. A means have juice spores. B perforated septa. Perforated septa means ascomycetes. So answer should be second one. Third is glomerulus. This is here second, third, first, second, third. All has third choice of glomeromycetes. And D is the have juice spores with flagella. Juice spore flagella means chytridiomycetes. So answer is second. Now this is the December 16 question, and you can solve by this material easily, very easily. This is the answer of this previous questions of adrenal gland. You can check your answers, and all the best.